All right, so this is our online lecture day that we're going to have covering Chapter 11, Cell Signaling. So you can watch it on YouTube. Please be aware that as part of your participation for this class, I'll have a worksheet that you need to download. I'll also have passed it out in lecture prior to this. So if you can't find it, go back to Canvas and download the worksheet. And you will need to hand that in next week when we have class, and that'll count for this day's participation. So it'll help you guide you along this lecture. So keep in mind that cell signaling is a very broad topic, very detailed. When you take cell biology, you'll spend many weeks doing cell signaling. So again, just like a lot of the chapters in Bio 1, we're going to cover just some overviews topics. So this isn't a long lecture, and so just stick to this lecture as part of your guide for studying. But there's a lot more detail in the book and a lot more examples of different pathways in the book that I'm not covering here. You're more than welcome to read about it, but I want you to focus on the broad concepts. All right, so the goal of this topic is to kind of introduce you to how cells communicate with their environment. So as you know, cells need to be able to communicate with the extracellular environment, and they do so through a general mechanism called cell signaling, which incorporates tons of different types of cell signaling. So one thing to keep in mind before we talk about the examples of cell signaling is essentially different ways cells can communicate. And so cells communicate by receiving signals, and these signals are typically small molecules that are usually released by cells themselves. So you'll have a signaling cell that will release a molecule, and then a receiving cell that will receive the signal. And so there's a couple ways that cells can do this. This is called local signaling. So this is a way for cells to signal to each other that are very close together. And so what you'll see up here is there are two connections between cells called gap junctions, and we didn't cover these as part of the cell structure, but these gap junctions are actually pores. These are ways that cells can attach to each other, and they're actually open to very small molecules. So cells can actually, one molecule can go to the other cell and back and forth. They can actually communicate that way. Plants can do the same thing. The gap junctions in plants are actually not called gap junctions, they're called plasmodesmata. And the reason is these incorporate the cell wall of plants. Oh, my apologies. These incorporate the cell walls of plants, and so they're a little bit more complex. But these are direct contacts between two cells. Another example of how cells can directly contact each other is right here. On this cell, this is the signal. So cells can actually have a signal that is part of their membrane. It's a transmembrane protein. And then another cell, the receiving cell, can actually have the receptor, right? And so this part of the cell is the receptor. And they have to directly contact each other in order to signal into that cell. So in this cell, the cells themselves are connected. In this cell, the transmembrane proteins of the cell are connected. And so that's an example of local signaling. And when types of signaling where there is not direct contact, this is actually called paracrine signaling. So this is still local. Paracrine refers to the fact that a signaling cell or a secreting cell will make a signal and secrete it to the extracellular environment. And then the receiving cells are very close by. Paracrine signaling is also essentially called neighboring, or neighboring cells are the ones that respond to it. All right. An example of paracrine signaling is also neuronal signaling, so called synaptic signaling, where the signal is released from a neuron and then a target cell is very close by. Both of them are examples of local signaling, just like the contact signaling we just covered. But this is called paracrine because the messengers are actually secreted to the extracellular environment. If you remember from this topic, the signal here is actually part of the cell, part of the membrane, and the signals here are intracellular, so they're communicating between two contacts of the cell. But in paracrine signaling, the signal is released to the extracellular environment. And then the fourth form, I guess, or the third form of signaling that we covered is long-distance signaling. So this is called endocrine signaling. And endocrine signaling is typically done by hormones. This is the main mechanism for hormones being secreted into your bloodstream. And so it's also referred to as hormonal signaling. But whenever we talk about hormone signaling, that's called endocrine signaling. And the way this works is a signaling cell or an endocrine cell will synthesize the signal in vesicles and then release the signal to the outside environment. But this signal is not just going to signal to neighboring cells. It'll actually go into the bloodstream and travel throughout your entire body until it binds to a target cell that has a receptor for that hormone. All right. And so this is long distance signaling. It takes the signals can travel from your brain all the way down to your foot. And so this is the longest distance that signals travel, called endocrine signaling. Regardless of the type of signaling, a cell has to have several components in order for cell signaling to occur. So one thing that's very important to understand is you can't have signaling without the signaling molecule. So at the start of any signaling pathway, some cell has to secrete a signaling molecule, or through the contact-dependent mechanism that we just talked about, has to have an internal signaling molecule. But all cells don't respond to signals, right? So in your body, you've got hundreds of different types of signals going on, or signaling molecules going on, but not all of the cells are responding to it. What makes a cell respond to that signal is whether or not they actually have a receptor. So if for a cell to even sense a signal molecule, they have to have a receptor. These receptors can take the form of usually transmembrane proteins that are sticking through the membrane to the outside of the extracellular environment so that they can bind to whatever signaling molecule is there. Sometimes, though, as I'll show you an example, these receptors can actually be intracellular. So they're not just membrane proteins, but they're inside the cell as well. But regardless of what example we're talking about, all cells have to have some mechanism of receiving the signal. And usually this is done by having a receptor that binds specifically to that signal molecule. And these receptors are unique. So this receptor, for example, would only bind to this molecule and not a different molecule. So they're very highly specific. All right, another component of cell signaling is once a signal has been bound to a receptor, that message that the signal is here bound to the receptor needs to be relayed to the inside of the cell. And so as you'll see from all the examples that we're going to do, cells have various molecules, and these are going to be very diverse. So diverse meaning there's lots of different ways the cell does this. But all of them are going to do the same mechanism. They're essentially transmitting the signal from the receptor to the inside of the cell somehow. And they can do a variety of things. I'll show you some different examples of how this occurs. But this process is called transduction right here. And so cell signaling actually is called signal transduction. And what it means is the ability of the cell to receive a signal and relay it into the cell so that the third component of cell signaling occurs. And what that third component is the activation of some response. Cell signaling's goal is to tell the cell to do something or not do something, depends on what the signal is. So at the end of the day, you have to receive it. It has to be transduced or relayed inside the cell to specific areas of the cell, and then it has to turn on or turn off, usually turning on, some type of cellular response. This 3 stages of cell signaling is called signal transduction, or we'll call it a signal transduction pathway. So for example, some of the examples I'm going to give you today are what are called signal pathways or signal transduction pathways. All right, so that's just what I talked about, the response. At the end of the day, when we look at cell signaling, we should be looking at some type of cell response to that signal. All signals have the cell respond to it. Okay, so here's a quick video to show you how this works.
Cells communicate with one another by means of chemical signals. For the receiving cell, there are 3 stages in the signaling process: reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the third stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes.
So we're going to look at all three of those stages in much more detail. So we'll look at reception, transduction, and cell response specifically. And I'm going to have to use certain examples because examples are the best way to talk about this. So we'll talk about reception. Reception happens essentially by receptors. So this is an example that you're looking at here. This is called a receptor. And receptors can be either transmembrane, all right? So these are these transmembrane proteins we've talked about a lot in class. These are the membranes that span, or sorry, these are the proteins that span the membrane. Or they can actually be intracellular receptors. So they can be proteins inside the cell that will respond to the signal. But the traditional cell signaling that you're going to be taught, most of your biology undergraduate, is going to focus on transmembrane receptors. So this is called the beta-2 adrenergic receptor. This is just an example of one type of receptor. You'll notice that it's got many alpha helices that are spanning the membrane. And then what's important is right on the extracellular side of the receptor, you'll notice here and here, are exposed binding pockets that will bind to the signal, whatever that signal is. All right. So in cell signaling, we call the signal the ligand, right? And then obviously the receptor is the receptor. So receptors always bind to ligands, whatever that molecule is, all right? When receptors bind to ligands, a very important thing happens, and what happens is the receptor actually changes shape. Now sometimes it's not dramatic, so this thing might just change a tiny bit. For example, this loop down here might, stick out instead of sticking down. And it's that shape change which is the event necessary to transduce the signal. So when ligands bind to receptors, they change the shape of receptors, and that is the first step of cell signaling. And this is just what I was talking about before. Most of the receptors are going to be plasma membrane proteins, transmembrane proteins. Be aware that not all. Some receptors are intracellular. They sit inside the cytosol and bind to signals there. So you may see some very specific examples of that, but the majority of receptors that involve cell signaling are plasma membrane proteins. So the best example I'm going to give you is the most common example of receptors in the cell, of how cells do cell signaling. In fact, the majority of cell signaling takes place through this mechanism. Not all, of course, but a lot of it does. And so this is a good example to learn how this works. So this is called G protein coupled receptors. All right, so this is a long word, but essentially what it means, it's a receptor, okay, obviously. And this receptor is a transmembrane protein, all right? And the reason why it's called G protein coupled receptors is that the receptor is linked physically to something called a G protein. And a G protein, as you see here, is a protein that's on the cytosolic side of the membrane, and it's associated with the membrane. So it's not a transmembrane protein, it's what we call a peripheral membrane protein. So it's bound to the cytosolic side of the lipid bilayer, and it actually floats back and forth. It diffuses throughout the membrane back and forth. All right. What's important is that in its inactive state, the G proteins are bound to a nucleotide called GDP, and they're bound to the receptor. So when a G protein coupled receptor has no signal, as you notice here, this is empty, then the G protein that it's linked to They interact with each other, even though this picture doesn't show that it's interacting, but it's actually sitting right next to it. And it's bound to GDP. Okay, so what's really important is that you understand how these G proteins work. So a GDP bound G protein is inactive. And what inactive means, it's not relaying the signal. So think of the G protein as one of these relay molecules. So there's no signal, the G protein is inactive. When a signal does bind to the G protein coupled receptor, this G protein will exchange. What I mean by exchange is it gets rid of GDP, all right? It picks up GTP from the cytosol. So there's a large amount of GTP in the cytosol just floating around. And so the G protein has very high affinity for GTP, but it needs to get rid of the GDP to do so. All right, so it won't just do this automatically. It has to wait for the G protein coupled receptor to signal to the G protein to kick out the GDP. It'll then bind to GTP, and now this G protein, you can draw little whiskers or something, this G protein that's bound to GTP is now considered active, which means that it'll then stimulate downstream signaling so that the relay or the signal transduction part of this is activated. Okay. What's important to understand is these G proteins are something that we call GTPases. Sorry about my handwriting. What GTPases are is they are enzymes. So this G protein has several roles. One of its role is to be a relay molecule for this signal, for this receptor. So it binds to G protein coupled receptors and it acts as a relay molecule. These G proteins are diverse. There's not just one of them, there's hundreds of them, and they all do different things, so we won't worry about what they do yet when they're active. But another function they have, in addition to their signaling function, is they have the ability to activate this enzymatic activity called GTPase. And what that means is that this G protein can hydrolyze GTP to GDP and PI. And this happens automatically. So it's kind of like an egg timer. It'll bind to GTP and become active. And after a few seconds, it'll automatically activate its GTPase activity. And it will hydrolyze GTP. And what that means is it breaks apart the terminal phosphate of GTP. And now it's bound back to GDP, and this G protein is now inactive again. So these G proteins cycle through this inactive, active, inactive, active state. And so the way this works is a signal molecule will bind to the receptor. The G protein will exchange GDP, get rid of it, and for GTP, bind to it. This makes it active. Binding to GTP activates what's called its intrinsic GTPase activity. What that means is it activates its built-in GTPase activity, and after a few seconds it'll hydrolyze GTP to GDP, and this G protein essentially turns itself off. All right? When it turns itself off, it'll go back to the receptor, and if there is no signal there, it'll stay just inactive. But if the signal is still at the receptor, it can turn itself back on again by the same exact mechanism. So these G proteins are these relay molecules that have kind of this built-in switch where they become activated by a receptor, they turn themselves off. If the signal is still there, they'll be reactivated so that the signaling can continue to occur. But if the signal is not there, then they stay inactive. All right, so it's kind of a complex regulation mechanism. Okay, so this is a better picture of how this works. And if there is no signal around in your cell, the G protein coupled receptor is empty, so the active site has nothing bound to it. That means the G protein is inactive, which means it's bound to GDP. You have to understand that G proteins are inactive when bound to GDP. All right. If a signal binds to the G protein coupled receptor, it'll then activate the G protein, and so you should stop and ask yourself, how is a G protein activated? A G protein is activated by exchanging GTP for the GDP it was bound to. This then activates the G protein. Now, what do G proteins do? Right? Now, I told you they turn themselves off, which takes a few seconds. In those few seconds, the active G proteins will actually relay the signal. Remember, these are relay molecules. So what the active G protein will do, a lot of times, is it will go and bind to some type of enzyme that is in the plasma membrane. And it will activate that enzyme. So you'll notice up here, in the absence of signal, this enzyme is not activated. So draw a big X out, X through it. So the enzyme does not work. In the presence of signal, the G protein is activated. That G protein then diffuses along the plasma membrane, binds to some activated enzyme. This is just an example of relaying the signal. And that activated enzyme actually does the cellular response. It makes something that the cell responds to, or that tells the cell to do something specific. So this is happening very quickly, very rapid, on the order of milliseconds. And then while this activation is happening, this G protein is going to then activate its GTPase activity, as we talked about, and it will automatically turn itself off. And there's two options. If it turns itself off and the receptor is empty, then signaling turns off. This enzyme is turned back off because the G protein is no longer activating it. If there is a signal, this G protein can diffuse back to the receptor, bind to it, and repeat this over again. So this can cycle back and forth over and over again and either activate the enzyme or sometimes only activate it for a few seconds, and that's all it needs to do to signal to the cell. Okay, so this is very important. This mechanism is one of the most fundamental mechanisms for how cells communicate that you should be able to explain steps 1, 2, 3, and 4. So go to this figure in your book and just read the figure legend and walk through it and use the terminology correctly, because it is confusing at first, but you need to understand how this mechanism works fully. Okay, so that's one example of the G protein coupled receptor. So let me go back real quick. Be aware, this receptor is called the G protein coupled receptor. This is the G protein itself. And then this enzyme is whatever that G protein activates. So this enzyme can do some cellular responses. And I'll show you an example of this at the end of the topic. But this is just one example of many of how cells do signal transduction, how cells relay a signal. And so I'm going to show you a second example for how cells transduce a signal. And it's actually a mechanism that cells use for lots of different purposes. So again, this is one of these fundamental concepts that you need to understand very well, because you're going to see it all throughout the cell, this mechanism, not just in the context of signaling, but also in other examples. So it's a very important mechanism. So one way that cells can transduce a signal, so they can do this step 2 right here, transduction, is to activate a relay molecule that is something called a protein kinase. All right, a protein kinase is an enzyme because it has ASES, and it's an enzyme that does an enzymatic reaction. I'm going to try and write it out long here. The enzymatic reaction that it does is called phosphotransferase. Okay, so protein kinases do phosphotransferase, right? And so what is phosphotransferase? Phosphotransferase is this reaction you see right here. It is the ability of an enzyme up here. So this is an actual protein kinase, a protein. And protein or enzymes, you remember, bind substrates, right? And so the substrate can be anything. It'll be any protein, right? It just depends on the example. So a protein kinase will bind to a protein, a target protein, not just randomly. These are very highly specific. And phosphotransferase means it takes the terminal phosphate of ATP and takes it off of ATP and transfers it to the hydroxyl functional group of 3 different amino acids. One of these 3 are going to get what's called phosphorylated. All right. The three amino acids that can be part of this reaction are serine, threonine, and tyrosine. Those are the only three amino acids that can be part of this enzymatic reaction. So protein kinases act on proteins by doing a phosphotransferase, transferring the terminal phosphate of ATP to the hydroxyl group of either serine, threonine, or tyrosine that is on these proteins. Remember, proteins are made up of 20 different amino acids, and these are three of those. And so all proteins can be phosphorylated if they have these three different amino acids in their polypeptide structure. And so what you get as part of this reaction is you get production of ADP, of course, because you've removed one of the phosphates from ATP and transferred it to the protein. And then the result is you get something called a phosphorylated protein. So let's talk about some terminology. This is called phosphotransferase. This is also termed phosphorylation. I know it's hard to see with my writing, but phosphorylation. Phosphorylation is the verb that you can use to refer to a protein kinase that does a phosphotransferase reaction. So this one word, phosphorylation, encompasses these two terms up top. So phosphorylation results in something called a phosphorylated protein, and that tells you that this protein has been chemically modified. It's sitting in the cytosol by itself like this, and then we can attach a phosphate to it. So what does phosphorylation do to a protein? Well, if you go back to the functional group lectures, I told you that phosphate groups, like this one, think of them as big, bulky kind of lightning bolts on a protein. These are highly charged, very large actually. And so what they actually do is they're going to change the structure of a protein. They're going to change the shape of a protein one way or another. It could be subtle, it could be dramatic, it depends. And changing the shape of that protein actually is going to change the function of the protein. Okay, so what will happen is when protein kinases are activated, they can be activated by a receptor. So let's make, for example, that this number 1 is a protein kinase. That protein kinase can be activated. That protein kinase will then phosphorylate a target protein. That target protein that is now phosphorylated has a different activity now. It may be an enzyme that does a function. It may be some other type of protein that modifies the structure of a cell somehow. It can have a lot of different outcomes. But think of this as a switch. A protein kinase is going to turn on a target protein by phosphorylating it. So it's a way of, again, relaying the signal inside the cell. All right. A couple things that I want to highlight here is this can occur over multiple steps. I'll show you an example of this, but this can you can activate one protein kinase, or sometimes multiple protein kinases can be activated, which I'll show you. This is also an example of something that's important about signal transduction. Signal transduction results in something called amplification of the signal. And what I mean by that is a single signaling molecule can activate this internal process of transduction, but one signaling molecule can possibly activate 10 different protein or 10 copies of the same protein kinase. So even though you have one signal, you now have 10 active kinases that are doing phosphorylation. So you're amplifying your signal inside the cell. And then more importantly, that we're not going to get into, but all of these steps, any process in a cell that involves multiple steps means that the cell can actually regulate it. It can control when these steps occur. It can control where these steps occur. It can control under what conditions they occur. So cell signaling is very complicated, not because these 3 steps are complicated. I would hope that you understand at least these 3 stages of cell signaling. But what makes it very complicated is the transduction step can be very complex and coordinated and regulated. But that allows our cells to be very specific and complex as well. All right, so here's an example of what's called a phosphorylation cascade. So here's an example of how a signaling molecule can activate a protein kinase, and through this step you can get amplification of a signal, and you can also get control of a signal or coordination of a signal. So this is called a phosphorylation cascade, and the way this works is you have a signaling molecule bind to a receptor. You guys all know that that should be step number one regardless. Step number one always involves a signal binding to a receptor. That receptor will activate a relay molecule. So for example, we already learned about G proteins. So let's imagine that this is a G protein. Okay, so this G protein is now going to be GTP bound and it will be active. Well, I told you that some G proteins can activate enzymes in the plasma membrane, but other G proteins can actually go and activate protein kinases. And so what they will do is they will bind to a protein kinase that's inactive, and they will make it now active. All right. So we have now transduced our signal from the outside of the cell through a G protein coupled receptor. The G protein relayed the signal, and now our effector protein, the protein that should get the response out of the cell, is an active protein kinase. This protein kinase is now going to go do phosphorylation, because this is what protein kinases do. They phosphorylate proteins. All right. So what happens in a phosphorylation cascade is this protein kinase doesn't necessarily just phosphorylate a target protein to get a response, but what this does is this phosphorylates another kinase, which activates it. Now we've got another active protein kinase. Well, this protein kinase will then phosphorylate another protein kinase, and now you've got another active protein kinase. So what's important to understand about what's called a phosphorylation cascade is you're getting two things. You're getting amplification, so amplify. Why is that important? Well, now if the G protein, let's imagine that this G protein that was active, all right, can activate 10 of these kinases. So now we've got 10 protein kinases active. Let's say each protein kinase can activate 10 of protein kinase 2. Well, 10 times 10 equals now, whoops, we're down here. We now have 100 protein kinase 2 active. And let's say that each protein kinase 2 can phosphorylate and activate 10 protein kinases 3. We now have 100 times 10, which now means that we've got 1,000 protein kinase 3s that were active. So from a single G protein, right, one G protein here, through this phosphorylation cascade, we have amplified the signal so that 1,000 protein kinase 3s have been activated. And this can happen on the order of milliseconds, so very quick. So this is an important component of cell signaling. How do you think about your fight or flight response? Why is it so fast? It's because the signaling pathways can amplify very quickly and get very large numbers of the signaling molecules with only small amounts of signal. What's more important as well is the coordination. So coordination. So active protein kinase 3 is going to hopefully go do our cell response, we think. All right? But sometimes these cell signaling pathways are so complex that protein kinase 2 will also do the cell response, or might do a different cell response. And active protein kinase 1, although phosphorylating protein kinase 2, can actually also do a different cell response. Right? So this is what I mean by coordination. The cell can actually do multiple things at once from the same signal, because we have now multiple active relay molecule or transducer molecules that can do multiple things at once. So we can amplify the signal, and then each one of these kinases can actually result in cell signaling. So as you can imagine, you might be confused by this, but this is why cell signaling can be very complex. For now, all I want you to understand about what is a phosphorylation cascade. A phosphorylation cascade is a signaling pathway using multiple protein kinases that phosphorylate each other, right? So active protein kinase 1 phosphorylates 2, 2 phosphorylates 3. And the reason why phosphorylation cascades are important is they can amplify a cell signal very quickly, and they can allow the cell to coordinate complex pathways or responses. So sometimes cell signals don't just result in a single response. Sometimes signals will result in multiple different responses in a cell, and this is an example of how the cell can do that. More importantly to this, you need to understand this. What does a protein kinase do? What does this phosphotransferase mean? What does phosphorylation mean? Because this is the thing that you're going to see over and over again in biology, is protein kinase is doing all sorts of things. The phosphorylation cascade is one example, but protein kinases do a lot more than just phosphorylation cascades. Okay, like I said, at the end of the day, at some point, these kinases should be acting on what we call effector proteins, or proteins that are actually going to do the cell response. So at the end of this figure, active protein kinase 3 phosphorylates some target protein, all right? And it can be anything. This is not a kinase, though. This is just some random protein. And by phosphorylating that protein, it has now made it activated, and that active protein will then go do some type of cellular response. All right, what's really important to understand at the end of this is, how do we turn off a signal? All signals need to have a turn-off mechanism. As you saw from the G protein coupled receptor example, G proteins are turned off through GTPase enzymatic activity. It's built-in GTPase that they do. The way protein kinases are turned off is actually from another enzyme. This enzyme is called protein phosphatases. Protein phosphatases, they're not shown here, but you could just draw it here. Whoops, I'm sorry. So they're not shown here, but protein phosphatases are also enzymes. So this is, I'll just say PP. This is a protein phosphatase. And what they do is they do the reverse of what protein kinases do. They dephosphorylate proteins. And what that means is they just remove the phosphate. So cut it off, and they revert the phosphate or the protein back to its original form. So protein phosphatases will basically turn off signals. So in this case, they're turning off this active protein. But you'll notice in their phosphorylation cascade, these PPs, these protein phosphatases, are also turning off these active kinases. So they're reversing the signal or turning off the signal that was occurring. And so be aware, or keep in mind, and don't confuse them, protein kinases phosphorylate proteins that usually turn things on. Protein phosphatases dephosphorylate proteins, which usually turns them off. All right, so we've covered two main concepts already. One is G protein coupled receptor signaling. This is an example of how cells use relay molecules to signal inside the cell. All right. Then I showed you protein kinases and phosphorylation cascades. It's a second example of how cells do transduction. Okay, so your book is going to give you lots of different examples, because transduction, although simply put is just relaying a signal inside the cell, it can take the form of many different ways the cell can do this. And so the goal of this topic is to introduce you to some very common ways that cells do transduction. So the third type of transduction, G protein coupled receptor signaling, is one. Protein kinase or phosphorylation cascades is a second type of transduction. The third type of transduction is utilizing something called second messenger pathways. A second messenger is any molecule that is made in response to signaling that will then act as a second signal. Okay? So the most common second messenger in cells is something called cyclic AMP. Cyclic AMP is a molecule that is formed from ATP. It's an enzymatic reaction, and so the enzyme is called adenylyl cyclase. You need to know this name of this enzyme. All right? Adenylyl cyclase. And what adenylyl cyclase does is it's activated in response to signaling. So let me write that down, because this enzyme is not always activated, but it's activated in response to G protein coupled receptor signaling. In response to GPCR, that's the abbreviation for G protein coupled receptor signaling. So that implies a G protein is activated in response to a signal. Then that G protein gets GTP bound to it. Remember, exchanges GDP for GTP. When it's GTP bound, that G protein will go and activate enzymes. And one of those enzymes is adenylyl cyclase. And what adenylyl cyclase will do is it removes two of the phosphates from ATP. It removes these two terminal phosphates, which is why you see this PPI sitting here. And it cyclases or makes a cyclic form of this nucleotide called cyclic AMP. You'll notice that this phosphate now has two ester bonds between the fifth and the third carbon, right, of this sugar. This cyclic AMP is small, it's rapidly diffusible inside the cell, and so this will go into the cell and can do a lot of different cell responses and signal to the cell to do certain things. And so this is called a second messenger because the first messenger was the original signal that activated the G protein coupled receptor. The second message is the cyclic AMP that was a result of the G protein activating this adenylyl cyclase enzyme. Okay, so second messengers are essentially internal signaling mechanisms from an external signal. And so here's an example of that graphically. So this is using that G protein coupled receptor example. Signal binds, G protein binds to GTP, which makes it active. Remember that a G protein is a polypeptide. It's not just GTP that's doing this. It's a protein that binds to GTP. GTP is just a nucleotide. This is now active, and this G protein will then diffuse over to adenylyl cyclase and turn it on. And as long as the G protein is bound to adenylyl cyclase and activating it, adenylyl cyclase will convert ATP to cyclic AMP very quickly. This is nanoseconds, very rapidly. So even in the few seconds that the G protein is active, it'll make millions of these cyclic AMP second messengers, or if not millions, but thousands of them. Okay, so what do these second messengers do? Well, a characteristic of second messengers is that they are small molecules inside the cell. What that means is once they're made, they will rapidly diffuse inside the cell. And these second messengers will then bind to proteins that have binding sites for them. So there are a lot of proteins that are activated by these second messengers. And so the example I'm going to give you is a very common mechanism that the cell uses for lots of different purposes. But it makes cyclic AMP, and one of the proteins that responds or binds to cyclic AMP is a protein called protein kinase A. And one thing to keep in mind is A is referring to the A in cyclic AMP. So that's how we can keep it straight. The abbreviation for protein kinase A is essentially pKa. Protein kinase A. And you guys already know what protein kinase A does now, right? Protein kinase A is a kinase. So when protein kinase A binds to cyclic AMP, it is activated. In the previous example, we were showing you phosphorylation cascades as a mechanism for activating protein kinases. This is a different mechanism for activating protein kinases. Cyclic AMP will bind to protein kinase A, and when protein kinase A is bound to it, it becomes active. Now that the protein kinase A is active, it can do its job. Its job is to phosphorylate target proteins. And those target proteins are going to then result in some type of cellular response. All right? So we start with an external signal binding to a G protein coupled receptor. That G protein coupled receptor activates a G protein, and you should be able to explain what activates means. The active G protein then binds to adenylyl cyclase, which activates it. Okay, so when adenylyl cyclase is activated, what happens? It converts ATP to cyclic AMP inside the cell. The cyclic AMP now, as a second messenger, will bind to protein kinase A and activate protein kinase A. Active protein kinase A will then phosphorylate target proteins. So I guess we could draw phosphorylation over here. To elicit some type of cellular response. Okay, so let me show you a real example of the cyclic AMP pathway. And this is the example that I want you to memorize because it's a good example for how this works. Signaling pathways, I know, seem complicated, but in reality, what they are just directions or step-by-step directions. It'd be no different than if I asked you to explain, the turning turn directions for how you get home or how you get to a store. So the way to study for this, if this seems really confusing to you, you have to know what the verbs mean. So you have to know what I mean by G protein becomes activated. You have to know what adenylyl cyclase does. You have to know what it means for cyclic AMP to bind to protein kinase A and so on. But once you understand those concepts, these are just directions of how proteins are going to behave inside the cell. So this example is essentially one of the examples of the fight or flight response. The fight or flight response is a hormonal response using epinephrine. Okay, so epinephrine is secreted into your bloodstream very rapidly, and it binds to a G protein coupled receptor on the plasma membranes of many different types of cells. And in this example, we're going to talk about muscle cells. One of the outcomes of the fight or flight response is to be able to run very fast, or at least to run away or do something. In order to utilize your muscles that way, you need very fast availability of glycogen. Remember that glycogen is the usable sugar in our muscles, the energy storage that our muscles use. And so here's how this works. And what these numbers on the side are showing you are going to be the amplification steps. Remember, every step of this is amplification of the signal. So G protein coupled receptor binds to epinephrine, step 1. Step 2, what happens when a G protein coupled receptor binds? It activates a G protein, right? What does that mean to be activated? You should be able to explain what this molecule is right here that's bound to it. The G protein is active, and that G protein now goes and binds to adenylyl cyclase, which activates it. What does adenylyl cyclase do? Adenylyl cyclase converts ATP to cyclic AMP. All right, remember, small, rapidly diffusible molecule inside the cytosol that will then go and bind to inactive protein kinase A, so pKa, and now pKa is active. All right, so what does pKa do in this example? So remember, pKa can do a lot of different things in a lot of different signaling pathways. This is a common mechanism, but once you get to this point, it'll start to vary based on what the signal itself is. So because it's epinephrine, and because we want to get glycogen ready, what pKa does is going to activate two kinases, not directly, but it will activate a kinase cascade. pKa phosphorylates, remember all kinases phosphorylate. pKa phosphorylates an active phosphorylase kinase, and this is where it gets really confusing because the name of this kinase sounds like phosphorylate, of course. So pKa phosphorylates this, and then if you want on this picture, draw a big phosphate next to it to tell you that these are phosphorylated. Phosphorylase kinase is a kinase, so it's going to phosphorylate something. It'll phosphorylate an enzyme called glycogen phosphorylase. All right, and glycogen phosphorylase will essentially do this reaction. It'll phosphorylate glycogen to make glucose 1-phosphate molecules, which are ready to use sugar molecules that can go right into glycolysis and give us energy right away. All right. So what you get, this is the best example because it gives you many different types of signaling pathways all in one. It gives you G protein coupled receptor signaling. It gives you second messenger signaling. And also it incorporates a protein kinase or a phosphorylation cascade. So if you can understand what is happening in this whole pathway, you will be able to you have then fully understood what we have talked about, all the different examples individually. So as part of your worksheet, I'm going to make you write out these steps. And what I want you to write out is basically what I said previously. You can pause it and go back. Is epinephrine binds to G protein coupled receptor molecule. G protein coupled receptor molecule activates G protein. Tell me how a G protein is activated. Remember, it spits out, or GDP goes away, GTP goes into the protein and activates it. the GTP bound G protein, and that's a mouthful, activates adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP. Cyclic AMP binds to protein kinase A and activates it. Active protein kinase A phosphorylates an enzyme called phosphorylase kinase. Phosphorylase kinase phosphorylates an enzyme called glycogen phosphorylase. Glycogen phosphorylase phosphorylates glycogen, essentially. It doesn't really phosphorylate it, but it modifies glycogen so that glucose 1-phosphate is produced. Okay, so what I want you to focus on is the verbs I was just using, right? What is each of these steps doing? What's the do in that? Okay, so this is all taking place in the cytoplasm, right? Epinephrine's on the outside of the cell, but once it binds to this receptor, all of these molecules are being activated in response to that single epinephrine. You'll also see all the amplification steps. We've converted a single epinephrine molecule to 1 million glycogen phosphorylase molecules in the order of milliseconds. So this is an extremely rapid response, right? What's not being shown here, but what's an important concept about cell signaling is every single one of these steps can be regulated by the cell. There are many other proteins that are involved in here that actually can regulate when this occurs. So these are very complex pathways. And the last example I want to give you is not a specific example like epinephrine, but just to show you another type of cell response. So the epinephrine response is a very rapid response. And the reason why it's rapid is because the kinases are acting directly on glycogen in your muscle cells. So the transduction part is acting on something existing in the cell. Sometimes though, signaling molecules or signaling pathways aren't rapid, they're a little bit slower, and the reason is because sometimes the signaling molecules need to tell the cell to make something, or make more protein, or turn on genes, or turn off genes. And so many of these signaling pathways will utilize the same concepts that we just went through, right? Signal molecule, receptor, maybe there's a G protein there, phosphorylation cascade, but instead of activating a cellular response that makes glycogen or maybe turns on a protein immediately or just does something very rapid, instead these kinases will actually go into the nucleus and they will turn on an enzyme or enzymes called transcription factors. And you haven't learned much about transcription factors yet, but transcription factors are proteins in the nucleus that activate transcription of genes to make messenger RNA to ultimately make protein. So in this example, this signal isn't telling the cell to make glycogen or something like that, something quick. This signal could be something like a growth factor. And maybe it's telling the cell, hey, I need you to become bigger, or I need you to make more protein, or I need you to become a different type of cell, maybe grow more muscle or things like that. Growth factor signals will ultimately signal to something called transcription factors, because the transcription factors will then go into the DNA and turn on the genes that signal molecule is telling it to do. So for example, if we need to build more muscle, the first thing we need is more muscle protein, right? More actin, more myosin, those sort of proteins that we discussed. And the way the cell will do that is growth factors will turn on the transcription and translation of those muscle-specific genes. Remember that these are slower. Transcription translations can take anywhere from 6 hours to 24 hours, depends on the gene that the cell needs to make. So these are slow responses, unlike the epinephrine example that was a very fast response. And the reason for that is because ultimately these examples are turning on transcription and translation. So I show you this as an example of other types of effects that signals can have, but I also show it to you because I want you to understand what a transcription factor is. A transcription factor is a protein inside the nucleus that is activated in response to cell signaling. So it can be activated by being phosphorylated by a kinase, and that activation will turn on transcription of a target gene. All right, so your worksheet for today, that's all I have. Your worksheet for today Hopefully it's going to guide you through this lecture. I would recommend doing it while you're doing the lecture, but you can do it afterwards as well. You can, of course, go back and play any part of this lecture over again. And I want you to focus on mechanisms, what I call the how of something. How do G protein coupled receptors work? How do G proteins work? How do kinases work? How do phosphatases work? That type of thing. If you can understand that, then the rest of pathways are essentially putting them in certain orders or configurations, but putting them in different directions essentially. All right, keep in mind that you have no mastering homework or quiz this week. So no mastering homework due on Sunday. So you have this week off of mastering, and you will not have a quiz on Monday or Tuesday of next week. The only thing I have you do for this topic, because it's virtual, is to complete that worksheet. So please bring that worksheet on Monday when I see you again. I hope this was a useful topic, and I will see you all on Monday.